Hello everyone, my name is Emmalyn Leonard and I am a senior undergraduate in the Neo Lab in the Institute of Neuroscience. Today I'm going to talk about my work exploring the neural basis of visual processing. Visual input first hits the retina in the back of the eye, then travels to the brain via the optic nerve to brain regions including the primary visual cortex, or V1. Although V1 has been named after the single sensory modality of vision, recent work suggests neurons in V1 respond to non-visual signals as well, such as movements of the head, eye, and body. Work investigating this phenomenon is often significantly limited by the difficulty of recording neural data in a freely moving animal, and as a result, experiments often include head fixation of the subjects, leading to fewer movements in a less naturalistic experimental environment. Here, we've developed a novel technique for recording neural activity in the freely moving mouse while simultaneously recording head and eye movements. Our technique uses chronic multisite silicon probe implantation, as well as a 3D printed device holding a camera for recording the mouse's visual input, a camera for recording activity of the eye, and an inertial measurement unit, or IMU, which is all shown in the left of Figure 1. Our experiments are set up into two phases, the first being the head fixation phase, where we record visual responses of V1 neurons using previously published methods, which is shown in the middle of Figure 1. The next phase is the freely moving recording, shown at the right of Figure 1, during which we record from the same cells as in the head fixed condition, while recording the animal's head and eye movements as they are allowed to freely explore in an arena. We find individual neurons in V1 to be excited by specific movements, similarly to how they are excited by specific visual stimuli, suggesting the specific tuning of V1 neurons goes beyond their response to visual stimuli. So some examples of the types of data we collect are shown in Figure 2. On the left are frames taken from the worldview camera, the eye camera, and the top-down camera over the arena. On the right are example traces of data we collect during experiments, which are aligned to the spiking activity of individual neurons, shown in the raster plot at the bottom. So from the head fixed recordings, we generate receptive fields through spike-triggered averaging. Spike-triggered averages, or STAs, are averages of visual input, in this case stimuli on the stimulus monitor, present when a neuron fires an action potential. So from these averages, we can note specific regions of the visual field where the unit responded powerfully to stimuli, and these are known as receptive fields. Examples of these are shown in Figure 3, along with the individual neuron's firing rate as a function of contrast present in the stimulus. In Figure 4, we see the tuning of different individual V1 units to a grading stimulus, where each neuron shows peak activity to a specific orientation or spatial frequency of the stimulus. Now, from the freely moving experiments, we find individual neurons to be tuned to respond to compensatory head and eye movements, as well as saccadic eye movements. Compensatory movements are those used by the animal to maintain its visual focus while reorienting its head. Saccadic eye movements are rapid shifts in the animal's eye position that are not compensatory, meaning they accompany the head movements in the same direction and shift gaze. Examples of this tuning is shown in Figure 5, where we see two units tuned to only one direction of movement, and one unit that is responsive to movement in both directions. Receptive fields are difficult to deduce from freely moving recordings, as there is little control over visual input. But using a generalized linear model, we found that, when accounting for factors such as head movements and eye movements, we can calculate a receptive field similar to that found in the head fixed recordings. So in Figure 6, we see the head fixed STA, as well as the generalized linear model produced STA, and find that their features are highly similar. This exciting preliminary finding suggests that generalized linear modeling of receptive fields is an effective method to calculate receptive fields of V1 neurons from purely freely moving data. In Figure 7, we show the action potential waveforms of 3 individual neurons, which we calculate for every single neuron in our data. The shape and dimensions of waveforms depend on the cell type and location, and thus are a powerful tool for organizing individual V1 neurons into distinct cell type populations. So now that we have performed a powerful characterization of the individual tuning of V1 neurons to movements, we will next perform population-level analyses of these cells to determine widespread patterns of tuning, as well as the relationship between visual and non-visual signals in V1. We can also use this technique to explore the activity of different brain regions, as well as the neural correlates of important behaviors that require vision and movement. Thank you for listening, and please email me at epl@uoregon.edu if you have any questions or are curious to see more data. Thank you, and have a great day.